All right. Hi, everyone. This lecture is going to be part 3 of our chapter 11 from your MERB text, the nervous system and nervous tissue. So we already talked about the different parts of the nervous system, neurons, glial cells, anatomy of those, as well as the physiology of an action potential. So the last part we need to talk about are the neurotransmitters. And the neurotransmitters are the language of our nervous system. So at chemical synapses, where we have communication between your presynaptic axon terminal and the postsynaptic neuron, or you can have some sort of effector such as a muscle or gland. At a chemical synapse, again, remember the way that communication happens through chemicals diffusing across the synapse, binding to the postsynaptic cell receptors, and those chemicals are going to be the neurotransmitters. Okay, there's more than 50 neurotransmitters that have been identified. Most neurons, make 2 or more neurotransmitters. So you can have different types of signals being sent by neurons. So neurons can either have excitatory influences, inhibitory influences, different types of messages being sent by each neuron. And obviously, like I said in the previous chapter, we can have different frequencies of neurotransmitter release depending on how frequent our action potentials are. Okay, so neurotransmitters we can classify 2 different ways, by their chemical structure and by their function. So we're going to talk about all the neurotransmitters first, just by their chemical structure. So the first neurotransmitter to talk about is acetylcholine, which we've heard before when we talked about muscle physiology. This was the first identified neurotransmitter and the best understood neurotransmitter. Most importantly, it's released at neuromuscular junctions. Also, other places in the body, it's released on the autonomic nervous system and some neurons within the central nervous system as well. It's synthesized from acetic acid and choline by an enzyme, choline acetyltransferase, and it's degraded by acetylcholinesterase. Okay, so again, these neurotransmitters, we don't want them hanging out too long in that synapse, so they either diffuse away, are reuptook by the presynaptic cell, or degraded by an enzyme. So acetylcholine has a specific enzyme, acetylcholinesterase, that breaks it down. Okay. Next class of neurotransmitters are biogenic amines. So we have catecholamines, such as dopamine, norepinephrine, and epinephrine, and they're made from the amino acid tyrosine. And the indoamines, such as serotonin and histamine. So serotonin is made from tryptophan, histamine is made from histidine. Okay, for these, I really want you to just focus on the names and what type of neurotransmitter they are. So don't worry too much about how they're synthesized. It's a little advanced for this class. Okay. The biogenic amines are used widely in the brain. These play a really big role in emotional behavior and our biological clock. A lot of imbalances with the neurotransmitters dopamine and serotonin play a big role in mental illness. Okay, so you'll see a lot of the drugs to treat depression are going to be targeting your serotonin neurotransmitters. So SSRIs are selective serotonin reuptake inhibitors. Okay, and we talked about what reuptake is. So reuptake is when the presynaptic neuron takes up the neurotransmitter back into its axon terminal so that the neurotransmitter isn't hanging around in the synapse too long. SSRIs block that reuptake process, which leads to serotonin being present in the synapse for longer. so it can have a greater effect on the postsynaptic cell. So a lot of cases, people with a depression, they don't have enough serotonin being released naturally. So SSRIs let their body use the serotonin that they have more efficiently, right? Because if it stays in the synapse longer, it was going to have a greater effect. So if they don't have enough serotonin naturally, that will allow their body to use the serotonin that they have more. So it kind of makes up for the lack of serotonin. Okay, but most drugs to treat depression are going to target these neurotransmitters, especially serotonin. Okay, and we're not going to talk about it in this class, but drugs are going to mimic the effects of some of these molecules or cause the release of them, which can lead to imbalances. So things like ecstasy causes the release of serotonin. If your body's releasing more due to using these drugs, it can have a long-term effect because if there's too much of that neurotransmitter present, your body's going to sense that and stop producing and stop releasing it because it thinks it has too much. So a lot of times you'll see that people who are abusing some of these recreational drugs will actually lead to lower levels of serotonin being produced naturally without the drug. Okay, and we're not going to talk about in this class very much, but if you take like psychopharmacology in the psych department, you go through all of the different drugs, their effect, what neurotransmitters and systems they affect, and the long-term results of using those drugs, which is, I find very interesting, but again, this class is a bit over our heads here. Okay, the next class of neurotransmitters are amino acids. Amino acids make up proteins, right? So this class is kind of difficult to prove which proteins are neurotransmitters. There's some arguments about different proteins that may be neurotransmitters, may not. But the ones that are proven neurotransmitters, glutamate, aspartate, glycine, and GABA. Glutamate is a major excitatory neurotransmitter, so it's going to cause excitatory postsynaptic potentials, right, which is you're allowing sodium in for depolarization, right, exciting the postsynaptic cell. GABA is a major inhibitory, so it causes IPSPs, inhibitory postsynaptic potentials, and its function, its way of working is it allows chlorine into the cell, chloride ions, which is going to hyperpolarize, which is going to make it more difficult for the postsynaptic cell to become excited. Okay. Peptides or neuropeptides are just strings of amino acids that have diverse functions. So some examples of peptide neurotransmitters, substance P, which is a mediator of pain signals. Endorphins act as natural opiates, so they reduce pain perception. So things like beta endorphin, dienendorphin, and enkephalins, which you don't need to know that, but and then gut-brain peptides is the last type of peptides. So somatostatin and choline-cytokinin play a role in regulating digestion. So there's something called the gut brain. So we have a lot of innervation in the nervous system in our digestive system, and it really influences a lot of processes in our body. If you get to some higher level neuroscience classes, again, you'll talk about this in a lot more detail. But again, we're just going to skim the surface here. The next class are our purines. They're monomers of nucleic acids that are both in the CNS and PNS. ATP technically is a purine and nucleic acids that is considered a neurotransmitter now. Adenosine is a potent inhibitor in the brain. So caffeine actually works by blocking adenosine receptors. So if you're blocking an inhibitor, you'll have an excitatory effect. That's how caffeine kind of stimulates you, keeps you awake. And it can induce calcium influx in astrocytes, which don't need to know. We also have gases and lipids acting as neurotransmitters. So gasotransmitters nitric oxide, carbon monoxide, and hydrogen sulfide gases. They bind with G protein coupled receptors in the brain, which I'll explain in a few slides. I talked about, I think, G protein coupled receptors when we talked about chemistry. They're lipid soluble, synthesized on demand. Nitric oxide is really important in learning and formation of new memories, and brain damage in stroke patients. And also you see it in smooth muscle relaxation in your intestines. Hydrogen sulfide gases act directly on different ion channels to alter their functions. Okay, and then endocannabinoids are lipids that act on the same receptors as THC, what you find in marijuana products. The endocannabinoids are the most common G protein leaked receptors in the brain. It's lipid soluble, synthesized on demand. They do believe that these endocannabinoids are involved in learning and memory. A lot of these neurotransmitters are still, the functions are not widely understood. So there's a lot of research currently being done on endocannabinoids and cannabinoids and their effect in the brain. But they do think endocannabinoids are involved in neuronal development, controlling appetite, which makes sense. You know, munchies, if you ever watched a movie where someone smoked weed or something, and suppressing nausea as well. Okay, so those are the categories of lipids. And they have a great diversity of functions. So our functions can be grouped into effects and actions. So we'll talk about those. So effects. We already talked about this, but you can have either excitatory or inhibitory neurotransmitters. Again, excitatory are going to depolarize the cell, get it closer to the threshold, make it more likely to fire an action potential. Inhibitory are the opposite, so it's going to hyperpolarize the cell, bring it further away from that threshold, make it more difficult to trigger an action potential. The main inhibitory ones are GABA and glycine. I'm going to emphasize GABA here. You're going to see GABA mostly being an inhibitory neurotransmitter. Your excitatory neurotransmitter, the main one we're going to see are glutamate. And then acetylcholine and norepinephrine bind to at least two different receptor types with opposite effect. So you can have acetylcholine being excitatory at neuromuscular junctions, but you can also have acetylcholine being inhibitory in cardiac muscle. So some neurotransmitters can either be excitatory or inhibitory, and it depends on the receptor, right? So acetylcholine is also always going to be released from the presynaptic cell, right? But on the postsynaptic cell, so acetylcholine, you can have nicotinic acetylcholine receptors or muscarinic acetylcholine receptors. So depending on what type of receptor it binds to, it can either be excitatory or inhibitory. And again, that's a bit advanced for this class, but you should know that some neurotransmitters can be both excitatory and inhibitory, and that's a result of different receptors they can bind to. Okay, and then the actions of the neurotransmitters can either be direct or indirect. Okay, so direct actions are actions that we've kind of already talked about, where the neurotransmitter binds directly to an ion channel. So it's going to promote rapid responses by altering the membrane potential directly by opening that channel. Okay, so this is what we've been talking about in all of our examples. So when we have acetylcholine binding at the neuromuscular junction, it's directly opening that ion channel, so it's going to have a direct action. Okay, indirect actions, you have the neurotransmitter still being released, but instead of binding directly to a ion channel and opening it, it's going to bind to a second messenger. And typically we're going to see that being a G protein pathway. And we'll talk about that a little bit more in a minute here. But G protein pathways, since it's not a direct action, it's going to be a bit slower, but it's going to have broader, longer lasting effects, similar to hormones. where they kind of have a long-term effect, not such a instant, quick, short-term effect. So if you're activating a G protein, you're going to see the effect happening, for a longer period of time. It might take longer for it to be initiated, but it's going to stick around longer. Okay, so the biogenic amines, neuropeptides, and some of those gases like nitric oxide are going to work through G proteins. We also have something known as neuromodulators, which are going to be chemical messengers that are also released by neurons. They're not directly causing excitatory or inhibitory postsynaptic potentials, but instead they're going to affect the strength of the synaptic transmission. Okay, so they can influence the synthesis, release, degradation, or reuptake of the neurotransmitter. So if they're causing a neurotransmitter to be sticking around longer or broken down quicker, obviously that's going to affect the results that neurotransmitter has on the postsynaptic cell. It can also alter the sensitivity of the postsynaptic membrane to the neurotransmitters. So it can bind or affect these ion channels on the postsynaptic membrane and make them more sensitive or less sensitive to the neurotransmitter being released. And it can be released as a paracrine, meaning its effect is local. Okay, so these neuromodulators aren't actually neurotransmitters. They're not causing an EPSP in the postsynaptic cell, but they're affecting the neurotransmitter's effects. Okay, so they're modulating how well that neurotransmitter is going to work. Okay. So our direct effects, open ion channels, right? So we've talked about this, but these channel leak receptors, you have an ion channel that can either be closed or open, and its state is dependent on the binding of a ligand. And in this case, the ligands are going to be our neurotransmitters. Okay, so if our neurotransmitter, for example, is, in this case, let's say it's acetylcholine, our acetylcholine binds to that channel linked receptor, causes it to open, and that opening is going to allow an ion to flow. Okay, so we talked about acetylcholine allows sodium to enter the cell, which is going to depolarize the cell. Okay, so if it's going to be excitatory, you're going to see sodium entering. We've talked about that already. The opposite of that, if you have an inhibitory one, chloride entering the cell is going to cause hyperpolarization, or you can also have potassium leaving, causing hyperpolarization. Okay, that would be an IPSP, whereas the excitatory is an EPSP. And then the very last bit here is just the G protein receptors. And we're just going to kind of skim the surface on these as well. If you take an advanced cell and molecular biology class, you'll talk way more in detail about these, but especially this being a summer class, I'm not going to spend too much time. But G protein receptors, again, are going to be indirect responses. They're going to be very complex, slow, and prolonged, meaning their results are going to kind of stick around for a long time. They involve what are known as transmembrane protein complexes, so proteins that go through the membranes and are going to be a lot of different proteins working together. Okay, and typically it's going to be widespread metabolic changes. Okay, so G protein link receptors can have a number of effects. But what's going to happen is going to have a neurotransmitter that binds to the G protein receptor, which activates that G protein, and I'll show a picture of this in a second. That G protein then controls production of second messengers, such as things like a cyclic AMP, cyclic GMP, calcium, different things. And those second messengers can do a number of different things. So the second messengers can go on to open or close different ion channels, right? And that can change to difference in the potentiation of the cell. It can activate kinase enzymes, can phosphorylate proteins, and even activate genes and induce protein synthesis. So you can have a G protein that creates a second messenger that then will activate genes and create more proteins in the cell. Okay, so again, these effects are widespread and long-lasting. If you're creating new proteins, that's not something as simple as just, opening an ion channel and causing an excitatory postsynaptic potential, where you're having major effects on that neuron. Okay, so basically how it happens, the neurotransmitter, again, binds to the receptor that activates the G protein. And instead of having a direct effect, that G protein is going to go and do a bunch of other shit. So in this case, we see that G protein is going to activate this adenine cyclase, which is going to convert ATP into cyclic AMP, which is the second messenger. And in this case, the second messenger is going to activate enzymes, change membrane permeability. So it's going to go on and have other effects in the cell. Okay? So you're still going to cause, something to happen. It's just going to go through a lot of these steps. And that's what a G protein complex is. You activate that G protein, and that's going to have a lot of downstream effects that can change a number of different things in the cell. Okay. Again, for our class, we're not going to get too in-depth on talking about this and what all these things are. But if you take higher level biology classes, different cell and molecular biology, you'll learn all about this stuff. And it can get a bit confusing. Okay, but that is the end of our Chapter 11 lecture. Let me know if you have any questions.